Biology of aging is the single greatest risk factor for 9 of the top 10 causes of death in the United States. It gets about 0.5% of the federal budget for biomedical research.
Dr. Matt Kaeberlein on the podcast. He is a world-leading scientist in the field of aging and longevity, as well as a professor of pathology at the University of Washington. He's the CEO of OptiSpan and co-founder of the Dog Aging Project.
If aging was defined as a disease, it would not change at all the clinical trials that you would have to do right now. Might it change popular opinion, the way people think about aging, the way policymakers allocate resources? That's possible.
What's your thoughts on the current state of biological aging testing?
People will say, all the clocks are different. Maybe one of the clocks is better than the others. Measurements, they're either correlating to chronological age or to your mortality risk, your risk of dying in the next 5 or 10 years. And so different algorithms are going to be correlated. Where the disconnect happens is in understanding what those algorithms are actually measuring.
So what are some of your top biomarkers to look at for longevity and health status?
Insulin is something that we don't typically measure. Extracellular matrix changes is one of the new hallmarks. And even if we could fix all of the hallmarks of aging, we might have significant increases in lifespan and healthspan, but we would still age.
So today I am so excited to have Dr. Matt Kaeberlein on the podcast. He is a world-leading scientist in the field of aging and longevity. He's the CEO of OptiSpan and co-founder of the Dog Aging Project, as well as a professor of pathology at the University of Washington with over 250 scientific publications, and his work bridges cutting-edge research and practical strategies to help people live longer, healthier lives. Welcome to the podcast.
Thank you. It's such a thrill to be here.
Yeah, I'm so excited to have you. There's going to be so much fun stuff to get into with you. Debunking some longevity things. I just walked you around my house and showed you a whole bunch of therapies that you probably are still waiting on the, significant studies to come out. But, we'll get into all that. And I'm going to be on your podcast too. So we can even talk about it more. So super excited to have you here. I want to kind of first start high level. So you were doing a lot of research on more like yeast and nematodes. Now you're doing it more so in dogs. Have you found anything surprising, to come over from one to the other?
Well, I would say, when I started, which was as a graduate student, so we're talking now the late 1990s. I've been in this field for a long time. At that time, we didn't, as a field, I think, know to what extent would the biology of aging really be shared across even mammals, let alone simpler organisms like nematode worms or single-celled yeast. And so it was an open question. At the time I started in the field, to be honest, I was pretty young. Aging wasn't personal yet. So I didn't, certainly I recognized the potential for what we were studying to the impact on human health, but I was really just more interested in the complexity of the biology and just a fascinating problem to study. And so, I wasn't really concerned about whether what we were learning in yeast would completely translate through to animals. When I became a postdoctoral researcher, so that's after I completed my PhD, then I started getting frustrated by it. It was kind of funny in the field at that time. This would be the early 2000s. The people who were studying aging in humans would say, you can't learn anything about aging from studying aging in a mouse. And the people studying aging in a mouse would say, you can't learn anything about aging from studying aging in a fruit fly or a worm. And the fruit fly and the worm people didn't have anywhere to go but to pile on the yeast people. So it was like everybody's just trying to justify, their research, but nobody was really trying to answer the question, to what extent is aging biology actually conserved? And so that's one of the things that I set out to do as a postdoc. And so this was when I was collaborating with Brian Kennedy, and we really tried to be as comprehensive as possible and look at every gene that had been shown to affect lifespan in yeast and every gene that had been shown to affect lifespan in C. elegans, the nematode worms, and just cross-reference them and ask, to what extent is that biology overlapping? And the fortunate answer is that it overlaps a lot. And I think what we've learned since then is that most of aging biology, we can think of it through the lens of, say, the hallmarks of aging, or we can talk about specific genes or pathways. Most of aging biology is shared, at least across the animal kingdom, and certainly very, very shared across mammals. So going from mice to dogs to people. I think So that's, I don't know if that's been surprising to me, but I think I have been surprised by how conserved the biology of aging seems to be. Certainly worth saying, not everything is the same. And I think the way I think about it is the underlying biology of aging is very universal, but the
Mhm.
way that manifests itself in terms of functional changes that go along with aging, specific age-related diseases, cosmetic changes that go along with aging, the way that biological process manifests itself is different when you look in different animals. And that has to do with their unique evolutionary traits, right? The way they've
Yeah.
evolved, their environment that they're living in. And so that's where I think, we do need to start to think about, as we're thinking about therapies and interventions, we really have to be thoughtful. The same intervention may slow aging in a mouse and a dog and a person, but the outcome could be very different if that particular therapy has an unanticipated consequence, say in a person that you don't see in a mouse, because the mouse's aging trajectory is just different. So I'd say
Yeah.
that's one thing that I've been a little bit surprised about and also think a lot about. The other thing I would point to that has surprised me in the field is how malleable aging is, even late in life. So this
Mhm.
is something when I was a graduate student, a postdoc, myself and most people in the field, if you'd asked them, would have said, if you really want to have a big impact on longevity or health span, you have to start early in life.
Mhm.
That the returns are going to be diminishing the later you go before you start the intervention. And by intervention, it could be a drug like rapamycin, it could be lifestyle intervention.
Yeah.
So I'm using that category broadly. I think what we've learned in the last 15 years or so, and really the first demonstration of this was the mouse rapamycin study from 2009 when they started treatment in middle age. What we've learned is that you can have large impacts on both health span and lifespan, at least in some cases, by
Mhm.
starting interventions at, say, the mouse equivalent of a 60-year-old person, or the dog equivalent of a 60-year-old person, or the human equivalent of a 60-year-old person. You can have big impacts on health span, and lifespan starting late in life. So that tells us that the underlying biology of aging is much more malleable, I think, than we thought it was 20, 30, 40 years ago.
Yeah. That's so exciting. Do you have any, stories maybe of like who's the oldest person that you've seen implement interventions or, even in the lab?
Yeah, so this isn't one that comes to mind immediately, isn't someone who I know personally, but it's a story that I read about, and we covered this on the podcast. There was a gentleman who I think he's in his 80s now, and he's a world-class rower. Like, he wins all of the competitions for rowing in his age group. And my recollection is he lived a pretty sedentary lifestyle. I don't know how unhealthy he was,
Mm.
but he didn't row. He wasn't really exercising, wasn't paying a lot of attention to his diet until he was in his 60s.
Okay.
So, that's pretty impressive to go from, really not focusing significantly on your health to now being the world's best rower in your age group,
Yeah.
starting in your 60s. Another example I would point to is and this is more of a scientific clinical trial sort of data set, the work that Joan Manick did when she was at first Novartis and then Restore Bio. So they were looking to test whether a derivative of rapamycin called everolimus, or RAD001, could improve immune function in healthy elderly people based on
Mm.
its ability to modulate the biology of aging. So they took people who were between 65 and 85 years old, regular people. These are not biohackers. They're not, people who are paying a lot of attention to their health, just the typical general public. And gave them either placebo or everolimus for 6 weeks, and
Mhm.
then measured their body's ability to respond to a flu vaccine. And what they found was that in these 65 to 85-year-olds, just in that short window, they could actually improve the ability to respond to a vaccine. So again, I think, I certainly wouldn't want to suggest that we can reverse aging, or that we can, have dramatic effects on lifespan and health span for everybody between 65 and 85. But I also feel pretty confident that we can improve aspects of health
Mhm.
significantly for pretty much everybody between 65 and 85. And again, the way I really think about this is, until a disease pathology reaches the point where it has sort of become different from or outpaced the biology of aging,
Yeah.
we have the opportunity to target the biology of aging to improve health span and hopefully lifespan as well.
Yeah. Yeah, that's super important. And I think, it feels like I still get quite a bit of comments online of people thinking that it is too late for them, right? Or that they
Sure.
didn't start earlier. So I think it's a
Yeah.
really important, it was going to be a question if you didn't bring it up for you, like how late can you start? And basically it's never too late, right? Like
That's right.
today's,
I mean, that's certainly what the science tells us is that there is never a point that has been identified yet where it is too late to improve your health. And that targeting the biology of aging, I think, is the most effective way to do that. Now, people who aren't in the space might be like, what do you mean by targeting the biology of aging? I think one way to think about this is, I think you and I, correct me if I'm wrong, but I think we probably would align that lifestyle factors are the biggest levers you can pull to improve your health.
Definitely.
And there's a reason why eating a healthy diet, exercising regularly, getting good sleep, having good relationships improves not just one disease risk, but basically all of the diseases that kill people.
Yeah.
It's because those interventions do modulate the biology of aging, and we know something about those mechanisms. So yes, it is never too late. And the best way to do that for most people, unless you've already got those things dialed in, is to focus on improving your lifestyle factors first. I don't want to minimize the importance also of getting good healthcare, getting comprehensive diagnostics, looking at your biomarkers, because you want also to understand whether or not there are pre-existing problems that you need to fix, because you don't want to short circuit your opportunities to live as long and healthy as you possibly can because there's some underlying condition that you didn't know about. So I think those things go hand in hand. But again, getting back to your original point, I think for nearly everyone, no matter where you're at, there are steps you can take to put yourself on a better health span trajectory.
Yeah. And I feel like in most cases, of course, there's a few outliers, but you're also going to feel better right now, right? Like if you start working out, there's never a workout I regretted. So
Yeah.
I always feel better after.
I agree with you as well, although I do think for people who depend, that is somewhat dependent on where you're at in your journey, right? So
Yeah.
I think if you're already relatively healthy, then that is going to be your experience. If you have not focused on health, and you are starting from a place where it's just much harder to get up and move around.
Yeah.
The habits are harder to change. Maybe your, home environment is not conducive to healthy diet or quality sleep. I agree with you in principle, but I think for a lot of people, they don't get those immediate returns. And that's why I think for a lot of people, it becomes challenging to stay on that improved health span trajectory. So, and that's actually, maybe we'll talk a little bit more about things like the GLP-1 drugs. That's where I think those kinds of interventions can potentially play a big role. And that's a double-edged sword for sure. But I think they work. There's no question about that.
Yeah.
And so I think for people, maybe as an example, who have struggled year after year after year to try to adopt a healthy diet and just can't stick with it, a drug like that can give you that feedback, because you're gonna see the results, you're gonna feel the results, and it can kind of kickstart it. And then when you get to a point where you've reached a psychological state, a physical state where you can get that immediate feedback from a workout, for example, then that's sort of self-perpetuating. So I agree with you in principle, but I also don't want to minimize the challenges that I think a lot of people face because they don't get that immediate gratification. And look, we live in a society of immediate gratification, right? Totally. So it's hard if you don't feel better, if you don't look better right away. And the reality is, while I do think you feel better right away after a workout, it takes a while before you're going to see the difference when you look in the mirror. Yeah.
Okay, well, maybe we'll say at least prioritizing sleep, right?
Yeah.
You got to feel better after a good night's sleep, right?
Absolutely. Yeah. I think sleep is again, one of what I think of as the 4 pillars of health span. Again, prioritizing sleep can be easier said than done for some people. I'm pretty fortunate. I don't struggle too much with falling asleep and staying asleep, but I know lots of people who do. And I think that's a place where, if I think about nutrition, exercise, sleep, connection. I think depending on your specific situation, sleep is the one where it can be hardest to really dial it in. Because I think some people, for a variety of reasons, whether it's stress or it's genetic predisposition that just makes it harder for them to sleep, or something about their home environment, it can be hard to know exactly what to do to really get good quality sleep.
Yeah, it could be so many things, right? So it's hard to eliminate all the things.
But we do know some of the big levers, right? I think alcohol is one that I don't think anybody can debate that alcohol degrades sleep quality, especially if you have alcohol late in the day. And I'm not one of those people who says you should never drink. I drink periodically, but I certainly drink a lot less than I used to.
Yeah.
And so that's one thing that we know what to do there. If you are experiencing a lot of stress and you wake up with your mind racing and you're thinking about things, trying to think about ways to minimize that stress can be helpful, but that's where it starts to get mushy. Like, how do you do that? Like, it's not easy.
Yeah.
If you're worried about something, it starts with recognizing what you're worried about, but it's not always easy to address that.
Yeah. Well, I definitely want to talk about GLP-1s in a sec, but while we're just talking about the biology of aging, so there were like 9 hallmarks, right?
Yeah.
Then 12. Now are there 14?
Something like that. Keeps growing. Yeah.
Yeah. So, which ones are you operating on? Just a quick overview.
Yeah. So there's a couple things I would say about the hallmarks of aging. So the hallmarks of aging, for people who maybe aren't aficionados, are really just a set of, I would have, a year ago, 2 years ago, said molecular processes, but even that's gotten blurry now. A set of processes that seem to be important for biological aging across the animal kingdom, and in most cases, all the way down into very, very simple organisms like yeast. And they include sort of fundamental types of damage like DNA damage or epigenetic dysregulation or mitochondrial dysfunction, chronic inflammation. So it's those kinds of things that are, at least in principle, associated with aging and causing some aspect of the functional declines or disease risk that goes along with biological aging. And as you alluded to, it started at 9, it went to 12. I was pretty happy with the 12. Then it went to 14 or 15. And it feels to me like it's starting to get a little bit, I don't want to say ridiculous, because all of the 14 or 15 are legitimate, but where do you draw the line? I think it's gotten blurred as to what actually is a hallmark of aging. Now it's just a collection of stuff that goes along with aging. The other thing I would say about the hallmarks, though, is, well, 2 things. They're incomplete, and we can come back to that. I sort of say, I don't know whether the hallmarks represent 5% of aging biology or 80% of aging biology, but my intuition is it's more like 5%. And that would mean there's 95% we don't know.
Yeah.
But they are incomplete. I don't think anybody would argue with that. And one of the implications of that is, even if we could fix all of the hallmarks of aging, we would still age. We might have significant increases in lifespan and health span, but we would still age. We would still have functional declines. We would still die. So
why would that be, though? Let's say if we fixed all of them, so we would have to continue aging. What's the reasoning behind that? Well,
if the hallmarks are in fact an incomplete picture of aging, we haven't fixed all of aging. Okay, right?
Yeah, that makes sense.
So first of all, I think we could, as a field, we could have a debate again about whether we would see, 3-fold increases in lifespan and health span, or 20% increases in lifespan and health span if we fix the hallmarks of aging. I don't think anyone who really is in the science would argue that the hallmarks are a perfect representation of aging, or that they encapsulate all of aging. Yeah. And I mean, as an example, we've added new hallmarks just in the last couple of years, right? Like extracellular matrix changes is one of the new hallmarks. Super important. And so you could fix everything that goes wrong, with mTOR and mitochondria and stem cells. If you don't fix the extracellular matrix, you're still going to run into problems. So the question would be, what don't we know about yet? And one piece of evidence that would support the idea that we've got a lot to learn is that, as a field, again, if you just look in the literature, we have not been successful at sort of incrementally or continuously being able to get larger and larger impacts on health span and lifespan, even in laboratory animals. Yeah. Really, we've been sort of stagnated for the last 40, 50 years in mice in terms of how far can we push longevity and health span. We've learned a lot about the mechanisms that underlie the biology of aging, but we really haven't been able to move down the field in terms of how effective our strategies are for ameliorating aging. So anyways, we started this with the hallmarks. The other point I wanted to make, though, and this is something that I think a lot of people don't think about, is the hallmarks aren't acting in isolation. There is a network, there's a biological network that underlies these hallmarks. And we know something about the connections in that network. We know some of the nodes in that network. So the nodes would be things like mTOR or AMP kinase or FOXO, the proteins that we know we can turn up, turn down, move around, and have an impact on all of the hallmarks at the same time. It's that network that I think we really need to be studying and fleshing out. Sure, it's great to look at the hallmarks, and they're really useful for giving us potentially targets to go after therapeutically or for communicating the biology of aging to people. But if we really want to have the largest impact we can on slowing aging, reversing aspects of aging, improving health span and lifespan, we need to understand that network, because the nodes that we work with right now, we aren't tweaking those nodes. And by that, I mean like rapamycin is an inhibitor of mTOR. It's a node in this network. We aren't tweaking those nodes because we've done a sort of a rational design approach to how best can we slow aging. We're tweaking those nodes because they came out of, unbiased genetic screens as the biggest signals that we got for lifespan extension, which is great. I'm all for, and I think we should go back to doing those kinds of unbiased genetic screens to find what we don't know. But the limited understanding of this network, I think, is keeping us from really identifying what are the most effective ways to go after biological aging while minimizing the risks of unanticipated side effects.
Yeah, that makes a lot of sense. Is part of the reason why we're moving so slow, do you think, is because lack of funding? What does the funding landscape look like right now?
Yeah, I think there's two big reasons. I think lack of funding is one, and I certainly can talk about that and talk about numbers. I think the others is an unanticipated consequence of the hallmarks of aging. So when the hallmarks of aging were first put forth in 2009, that had a huge impact on the way the field thought about the biology of aging. Suddenly we had these things we could point to. And so it got to the point, it's still this way, that it is almost impossible to get funding to study anything that doesn't fit within that framework, right? Yeah. And so I think there was a premature narrowing of the breadth of research in the field, in part due to the hallmarks of aging. The other reason why the breadth of research has been narrow is the lack of resources. And so it's been hyper competitive for resources in the field for a long time. It continues to be. There's this, I think, misperception out there that a lot of money has flooded into the field because of billionaires, right? You hear this all the time. That's not true. That's not to say that there hasn't been resources coming into the field because of large initiatives. The couple that I can point to that, have actually allocated significant resources are Calico, which is the company that Google started about 15 years ago now, and Altos, which is the company that Jeff Bezos and several others started, I don't know, maybe 5 years ago now. Yeah. But so far, those two initiatives haven't really led to any big progress in the field, and they're in the order of a few $1 billion. So just to give people context, the National Cancer Institute, that's one of the institutes in the National Institutes of Health, which is the largest biomedical research funder in the United States and still in the world, I think, it was about $6 billion a year. It's probably more than that now, maybe $8 billion a year. The biology of aging gets about $350 million a year. Wow. Biology of aging is the single greatest risk factor for 9 of the top 10 causes of death in the United States. It gets about 0.5% of the federal budget for biomedical research. So there's in my opinion, there's a huge misallocation of resources. And that absolutely has led to slower progress than could have been accomplished. I would say, I think there's been a lot of progress given the lack of resources that have been put into the field, but certainly things could go a lot faster if resources were allocated appropriately, in my opinion.
Do you think that and maybe, set the record straight. So it's not currently identified as a disease set, right? So if we did, that's obviously a big buzz. If we labeled it as that, would we get more funding? Would there be more initiatives? Or
maybe.
Not really?
Yeah, I don't know. It's a good question. I don't know whether a sort of formal labeling of aging as a disease would impact funding. It might, certainly. I think the argument that I've heard most common for people who want to call aging a disease is around the regulatory environment and the idea that if aging were a disease, then FDA could approve a drug for aging. That's a complete myth. Labeling aging as a disease will have no effect on getting drugs approved by FDA. All FDA really cares about is that you can show quantitatively that you can improve at least one endpoint that's relevant for quality and quantity of life for people, and that it's safe, right? So it's got to meet those two criteria. If aging was defined as a disease, it would not change at all the clinical trials that you would have to do right now. Right now, you can get a geroprotective drug, a drug that targets aging, approved by FDA, if you can show that it's safe and that it moves the needle for one relevant endpoint by improving quality or quantity of life for people. That would be true even if aging were a disease. So that changes nothing. Might it change popular opinion, the way people think about aging, the way policymakers, funders allocate resources? That's possible. I don't know the answer to that. I do think, one of the reasons I can point to two reasons why biology of aging has been underfunded historically. I think they're both legitimate in the sense that I understand why people feel and act the way they do. I also believe that it is counterproductive because it's a misallocation of resources. So one is, many times the way resources are allocated in the US federal government is because, somebody high up in politics actually makes a large effort to get funding allocated to a certain area. Yeah. And, everybody who has lost someone important in their life will point to something that they lost that person from. And sometimes it might be aging, like, oh yeah, died of old age. Much more often it's going to be, my mom died of cancer or Alzheimer's disease or heart disease. And so there's this personal connection with that disease that took somebody from you, right? And so I think that is why we often see, wealthy donors donate to hospitals for certain types of research, for disease-specific research, and why in the past, large legislative initiatives have gone towards, war on cancer, right? So I get it. What is missed there is that, well, it was really the biology of aging in most cases that caused that person to develop cancer or heart disease or dementia or something else. But that, I think, is part of the reason why resources have been misallocated. The other reason, and this has changed, is that I think historically the impression was you couldn't do anything about aging. So why would we put, large sums of resources towards something that we can't actually modulate, improve, right? The mandate of NIH is to improve human health. And so if the perception is that you can't improve human health by slowing aging, then it doesn't make any sense to put resources there. I think that thought was outdated 30 years ago. It's even more outdated now. I mean, it's almost ridiculous if anybody actually believes that we can't modulate aging today, because it's been proven over and over and over again. But I think still in some people's mind, curing cancer, curing heart disease is going to be an easier goal than slowing aging. I actually think it's completely the opposite. Everything I know about biology, and in the last 30 years, I've learned something about biology, tells me that slowing aging is way easier at the population level than curing cancer or curing heart disease. We can cure individual people's cancers. I don't think we're ever going to cure everybody's cancer.
Yeah, that's a really good point. Well, great. I'm glad we got to, kind of just have like a framework of what's going on in the longevity and anti-aging industry. So now I want to get into some testing. So I found it really interesting. I listened to this a while ago. You did a roundtable with a few other individuals, I think Peter Attia, and you guys were talking about biological age testing.
Right.
And I saw that you just made a post on Instagram about it as well, showing your data, right? So you did several, please tell the story. You did several different tests and then got a bunch of different answers. What's your thoughts on the current state of biological aging testing and where it might be going in the future?
Sure. So I'll start with the self-experiment that I did. But I do want to say, even before we dive into this, I think it's really important to distinguish whether we're talking about biological age assessments in the research world versus the direct-to-consumer world, because they're very different. And I sort of, had become a little bit wary of the direct-to-consumer biological age tests. And in this case, we're talking specifically the epigenetic tests, because we've been using them clinically at OptiSpan, and the numbers seemed all over the place to me. Just looking at the people in our programs and the numbers they were getting and the longitudinal values, and that was all with one company. But the numbers seemed off. I'll just put it that way. And so I thought, somebody really needs to just do the experiment where you, with the same samples taken on the same day, compare multiple tests from the same company and then across companies. And I don't know how many there are now offering epigenetic age tests, but it's got to be over a dozen. Yeah. So what I did was I purchased kits from 4 different companies. I got duplicate kits. In hindsight, I wish I'd done, triplicate, or the more is better, right? Yeah. But these things are also expensive. So I got duplicate kits from each company, 4 different companies. So a total of 8 tests. Yeah. And what I did was a blood draw, because not all the kits are blood. So some are blood, some are saliva. So a blood draw and then saliva sampling on the same day for all 8 kits, sent them back, and then just waited to get the results. And the results were, disappointing if you're somebody who hopes these tests actually are accurate. I don't know if it surprised me how bad they were as a class, meaning that I think I was 53 and a half when I did this. And the values I got back ranged from the low 40s to the low 60s. With a standard deviation of something like 7.5. Yeah. If you took all 8 tests and you took the average, it was pretty close to my chronological age. But, my interpretation as somebody who knows at least a little bit about statistics is this is useless from a clinical perspective. Yeah. So there's a few things to say, and people have said this, and there's some validity to it. Like, maybe one of the companies is good, and maybe that's true. How do you know which one? Yeah. Right? That really, I think, gets to the challenge here, which is that we have no way to assess precision or accuracy. Precision being how close are the replicate measurements to themselves, accuracy being how close is it to the true value. We can't assess either of those at all. Yeah. So that's a real problem, I think, in the direct-to-consumer biological age testing space. We also have no understanding of quality control, how the sample processing is being done. The companies are not transparent at all in that regard. There's no independent validation of the methodologies. And in my view, given all of that, they're just not actionable. The other thing people will say is, well, all the clocks are different. Maybe one of the clocks is better than the others because there's, and for people's understanding, when people say a different clock, what they really mean is a different algorithm for calculating this age metric based on the same data. So all of the data from these epigenetic clocks comes from looking across the genome at different sites of what are called methylation sites in the DNA. You can think of it as binary, 1, 0. It's either methylated or not. So you get this long string of 1s and 0s, and then you look for a pattern, a subset of those, let's say 100, that is most associated with whatever you're interested in your outcome being. Yeah. And so for the biological age, and I'm using the air quotes, measurements, they're either correlating to chronological age, which is easy, but not useful because I think we all know our chronological age, at least most of us do, to your mortality risk, your risk of dying in the next 5 or 10 years. And we can talk about how that's done. For now, let's just accept that it can be done. Yeah. Changes in health status or a specific disease. And so different algorithms are going to be correlated. Different sets of 1s and 0s are going to be correlated to one of those things. That's what the different clocks are looking at. One of the things I've heard people say recently is there's a specific algorithm called Dunedin pace or pace of aging, right? This is an algorithm, a set of 1s and 0s that is associated with change in health over time. So it's kind of like the slope of your health span curve. And I've heard people say, well, Dunedin pace is really the one that matters, and that's much more accurate than these other ones. And a question I have not gotten an answer to is, how do you know that? Like, great. It's great to say these things, but come on, this is science. Like, show me the data. There is no data that I'm aware of to suggest that Dunedin pace on the direct-to-consumer side is any more precise or accurate than any of the other measurements. And so unfortunately, in my data set, I only have 2 values for Dunedin pace because only one of the companies is licensed to use that test.
Yeah.
There are others that provide their own rate of aging metrics, and I don't know exactly how they measure them. So the 2 values were 0.81 and 0.91. So directionally good. I was aging supposedly more slowly than average, but still pretty far apart for things that were from the same sample. So there's just a lot of really, really bad misinformation out there. And I get more frustrated than I should by the misinformation, but it's because people are being harmed. That's what really bothers me. Yeah. So anyways, that's my rant on the biological age test. I just don't think the consumer tests are useful right now. I really don't. What I don't understand are the doctors who recommend these tests to their patients, because clearly there's problems with reproducibility. And I just don't think they're actionable. I think if you had an actionable test, and we have really good biomarkers that are really actionable. Yeah. That tells you something specific that you should change, right? Whether it's a lifestyle modification or a medication. I don't think these tests tell you anything in that regard. So that's the direct-to-consumer side. Yeah. That's where I would say I'm pessimistic right now. I think there are some things that could be done to make it a lot better, but the companies so far don't seem willing to do that. On the research side, these things are super, super powerful tools. So again, worth saying, it's a little bit unfortunate, at least from a scientific precision perspective, that these have been labeled as biological age tests. None of them measure biological age. They are all a correlation to biological age. And again, anybody who understands statistics will recognize when you start having multiple correlations, the errors propagate and it gets really problematic. So none of them are measuring biological age. I think it's better to say they're biological age estimators. But as I mentioned before, all of them are correlations. What they're actually measuring in most cases is methylation. In some cases, it can be facial features. So you can build these clocks from a lot of different types of data. You can take pictures of people and the AI can identify features in our faces that are correlated to chronological age or mortality risk or risk of cancer, whatever. So they're correlated to some type of data. Let's just say methylation with the epigenetic clocks. And it's then a subset of those epigenetic marks that are correlated to something else, mortality risk. Mortality risk is not biological aging. Risk of developing cancer is not biological age. It's correlated with biological age. That's why I say it's a correlation to a correlation. Okay. On the research side, these things are super powerful because for 2 reasons. I think 1, they give us potential insights into mechanisms of biological aging. If we can find specific methylation sites or groups of methylation sites that are regulating the expression of certain genes, and it's those genes that are causing mortality, that's really powerful. And that can lead to a better understanding of aging and therapies down the road. I also think if we can get this precision and accuracy piece worked out, these tools, as long as we understand what they're measuring and how accurate they are, will become really useful for clinical trials, as an example, stratifying patient populations for clinical trials, potentially even getting into clinical use once we know how accurate they are and what the error rates look like. The other place that I'm excited about these biological aging tools is the opportunity to not only get a single number, but to get estimates of specific organ and tissue function. And now this has been done with epigenetics, it's been done with proteomics, it's been done with transcriptomics, maybe even metabolomics. Again, multiple types of data you can look at, where from a single assay, you can get assessments of how well is your brain functioning and how well is your heart functioning and how well is your kidney and liver functioning. That can give us opportunities in individuals to identify early problems. And then we can go do additional diagnostics to assess, is there really something going on here? And then potentially do targeted therapies for that organ or tissue. That's where the actionability piece comes in. So I think there's a lot of promise with those tissue and organ specific multi-clocks. So there's lots of reason to be optimistic about these tools. The only real concern I have outside of people being misled and spending a lot of money on these tests that they could better spend to improve their health, is whether or not the backlash, when it becomes clear how bad these tests actually are, is going to damage the credibility of the field and damage the research use of these tools down the road. Again, maybe that's not going to happen. Maybe that's an unfounded concern. But I will say there are multiple examples where, hype and spin and backlash against that have led to delayed sort of regulatory adoption, right? And so as I mentioned, I think the promising features of these tools is use in clinical trials. That would be super helpful. But if the regulators think it's all BS because of all the noise, it's going to take a long time to educate them around that. So I do have some concerns about the way this is evolving right now.
Got it. The tools that you're using more in a clinical setting that you are excited and are saying working pretty well, it's a completely different system or algorithm than what is available to consumers?
You mean for the epigenetic tests in particular? Yeah, I mean, we're not using any in our clinics right now. The tools that are being used in the research world are also always evolving, right? That's partly why we have all these clocks, because somebody comes up with a better clock, a different clock that measures something different. So many of the algorithms that the direct-to-consumer companies use came from the research world, right? That's where they were first utilized. And so I think where the disconnect happens is in understanding what those algorithms are actually measuring, right? So again, what people call first generation clocks were correlated to chronological age, just number of candles on your birthday cake. Companies started rolling these things out as biological age tests, but it's just estimating your chronological age. That's not useful, right? I mean, I shouldn't say this. Most of the time it's not useful. So there's some disconnect with understanding what these tests are actually used for and how they've been developed. I think the bigger problem, though, I don't know where the error comes from. All I can tell you is in my experiment, there was huge error, and I've talked to that's not atypical. I've talked to lots and lots of people who've used these tests, doctors, consumers. The general rule seems to be the noise outweighs the signal. And we now know there are even circadian variations in these tests, right? You take the test at 9 a.m., you take it at 5 p.m., you're going to get a 10-year difference potentially. So I think the companies rush to market these tests. Where did we learn there's circadian variation in the epigenetic profile? From the basic research. But the companies were already selling these tests without knowing that, right? So I think there's that disconnect. And then I think the other problem is, as I mentioned earlier, there's no transparency around quality control. Some of these, and again, I know people at some of these companies, I like them, I respect them, they're well-intentioned. I believe they're trying to do it right. I know some people at these other companies who I don't respect, but we don't know. We don't know who's doing it right and who's not doing it right. And there's no regulation around this. Like you and I could decide we want to market our own epigenetic test and sell it on Amazon. We don't even have to process the samples, right? Who's going to know? That's the problem, I think, in the space right now.
Yeah, that makes a lot of sense. What about phenotypic age? People like kind of just plugging in some biomarkers that they have to get an estimate.
So again, I think it's important to understand that conceptually that's no different than the epigenetic aging clocks or any of the other aging clocks that you could use. The difference is which information, what data are you putting in to correlate to, let's just say health. I think, and again, if instead of calling these things biological age clocks, we call them health estimators, like I think that's a much more reasonable thing for what they're actually doing. And so, and this is what I was getting at where I said, I think there are biomarkers that are really actionable, right? So you can do this with, in fact, you mentioned pheno-age, that can refer to a specific algorithm that Morgan Levine published many years ago now. And in that particular paper, it was interesting. They found a subset of, it was 8 or 10 common blood-based biomarkers. If you just go get a common blood panel from your primary care physician, most of them will be on there. And you could correlate that to mortality risk. 10-year, I think it was 10-year mortality risk in that case. And in that same paper, they identified a subset of epigenetic marks that they could correlate to mortality risk. So in the same paper, you had 2 different algorithms using 2 different data types that are correlated to the same endpoint. So in that paper, there's a blood-based clinical chemistry, glucose, HbA1c, things like that. And and then there's an epigenetic algorithm. So they're fundamentally conceptually doing exactly the same thing. It's just the specific type of data that you're using to predict, right? So I'm actually quite bullish on these algorithms that utilize well-known clinical chemistry measurements. There's a really interesting one out of Singapore that Jan Gruber and Brian Kennedy and others have developed called Lineage. The first one was Lineage. Second one is Lineage 2. We're actually building that into our platform at OptiSpan because I think this is really where we should be looking. The reason why I say that is because the vast majority of the factors that make up this algorithm are actionable. They're blood-based biomarkers or things like blood pressure that we know how to do something about. Yeah. And so you can use this algorithm. You can say, okay, it's these 4 parameters that are having the largest effect on your predicted mortality risk. Lineage 2 is trained on mortality, but that's a reasonable thing to train on. Nobody wants to die. At least, I should take that back. Nobody who's healthy wants to die. And once you know what the factors are that are driving that mortality risk, you can do things to target those specifically. So you get to personalized, actionable information from this algorithm. Yeah. It's still, you still have to take a little bit on faith that mortality association is going to hold up in you. But I think it's a pretty good bet because these things make sense, right? Yeah. If you lower blood pressure in people who have high blood pressure, your mortality risk by the clock goes down, right? Yeah. So that's where I feel like this is the things we should be focusing on right now in the clinic, and let the research world take these tools, get them to the point where they are clinically actionable, clinically useful, and then use them in the clinic.
That makes a lot of sense. The other thing that I love is like phenotypic age, or you can even kind of do one with like ChatGPT. It doesn't cost any money.
That's right.
So again, if you're gonna, I've done biological age testing. I've done a whole variety of them, gotten different results. That's great. But I've honestly leaned way back from that because of, a lot of the research is saying that they're not that actionable. And I would rather, if someone is following me, buy a comprehensive blood panel to look at their cardiovascular health. How's your liver functioning? How is their everything? Yeah. And then take that data and do something with it. So.
Yeah. And I think that's a really good sort of contrast, right? So you could spend $500 on an epigenetic test. You spent $500 on a comprehensive blood test, you can get way more information that's much more valuable for you about your own health and things you can do to improve your health. Yeah. So it's just I think there are better tools. They're not as, sexy maybe as the biological age test, but they're much more useful at this point.
Yeah. And when you talk about mortality risk and how these tests could potentially be gauging to that, can you just go into a little bit more detail of what you mean?
Yeah, sure. So what we're talking about here usually is risk of dying over some time period in the future. So it's usually 3 years, 5 years, or 10 years. Okay. And the way these tests are able to be constructed is because we have what are called longitudinal data sets from people over the past 40 or 50 years. There have been several large human cohort studies, longitudinal studies of health or aging, that have collected samples from people 50 years ago, 40 years ago, 30 years ago, 20 years ago. Those samples are banked, so we have blood from those people. And we know what happened to them because they've stayed in the study over that time period. And some of them died. And so what you can do is you can take samples from 10 years ago, 20 years ago, measure the methylation status in those people, these sets of ones and zeros on the DNA, and make algorithms that predict give me the best 100 ones and zeros out of the million plus that we've got. Give me the 100 that best predict risk of dying in the next 5 years. That's basically what these tests do. So then that gives you the 100 methylation sites. That's what the kits are measuring. And then that'll give you a predicted value of your risk of dying in the next 10 years. That gets converted back to this age metric, which it isn't really, but that's what they call it, because we know across the population what the risk of mortality is across the population at a given age. So let's say the test predicts your mortality risk to be, 0.157%. That you're going to die in the next year. We can look at the population graph and say, okay, it's people who are this old who have that risk of dying, and that's your age. Mm-hmm. That's how these tests are getting from methylation sites to quote unquote biological age.
Got it. Well, thank you for that. And good to hear that the future could be bright, right, with these things. But right now, it's just,
They have so much potential. And so that's where, it's a balance between being frustrated some days with how they're being used and excited about where the research is going.
yeah. So, what are some of your top biomarkers to look at for longevity and health status?
Yeah, so we do a pretty comprehensive set of blood work as part of our clinical programs at OptiSpan. It's a pretty long list. It includes all the basic stuff. So maybe it's worth talking about the stuff that's not in the basic blood work panel. By basic blood work, I'm gonna talk about CBC Chem, glucose, A1C, basic lipids, things like that. So what's not in there? What don't people who only go to their primary care doc get that probably has the biggest bang for the buck? People have probably heard of ApoB, Lp(a). Those are both predictors of cardiovascular risk, certainly both known. Becoming more actionable. ApoB is actionable now. Lp(a) is becoming more actionable. So on top of that, for heart risk, I think one of the things that I have seen value in is the sort of more advanced lipid profiles that actually give you particle size in addition to just total numbers. What those things will give you is an estimate of your current cardiovascular risk, right? There are two things on top of that you want to do, right? One might be pharmaceutical, right? Maybe you want to do something actively, assuming you've tried the lifestyle stuff to lower your lipids. The other is that may suggest go get coronary CT, right? CCTA with contrast. That'll show you your hard plaque and your soft plaque. This is where I think it's important. It's useful for people to understand the difference between a blood-based biomarker, which can give you an indication of your risk, and something like an imaging test that can actually show you what is your actual plaque burden right now. So again, from the blood work side, I would put those in there. I think insulin is something that we don't typically measure. Everybody should be getting insulin when they're getting their fasting glucose, right? Because insulin sensitivity and resistance is really what is, I think, the big driver for metabolic health. Glucose is sort of the secondary marker that's got a lot more noise to it.
Do you like HOMA-IR at all?
Yeah, HOMA-IR, but you need your insulin to calculate HOMA-IR,
exactly.
right? So
I like looking at that value.
I agree.
Like as a woman, I think it's in general, but I like that value.
Yeah. So I would also say hormones is a big place where, again, you're probably not going to get it unless you have a problem that you perceive and you convince your primary care doc. So we do comprehensive hormone panels for both men and women. And
also, even if you do get it right, traditional doc is looking at it. I mean, the range is, what is it for men like testosterone? 200 to 1200?
This is why I said comprehensive. Yes, right. So just looking at testosterone, total testosterone is not sufficient. Even just looking at total testosterone and free testosterone is not sufficient. So you really want to get a comprehensive hormone panel that can really give you a picture, not only of where you're at, but if you are dysregulated, what might be going on. So we do comprehensive hormone panels at least annually. Again, depending on the person, it might be more frequent than that. We look at several different inflammatory markers, and this is where it starts to get, I think, a little bit complicated. And we're learning as we go. So we started by looking at multiple different tests for different types of autoimmune disease in order to understand, number one, how often is somebody who is asymptomatic going to show up on these tests? And number two, could they be used as a predictive or diagnostic outside of or within the reference range? Meaning that even if somebody doesn't get flagged as abnormal, maybe there are changes that you can see early on that reflect progression towards autoimmunity or just high chronic inflammation. So I don't want to name the specific tests right now because we're still figuring it out, to be honest with you. And I think there are a lot of inflammatory markers that could be looked at. And so I don't know what the best ones to use are. This is an area where there's also a lot of research happening right now. And so I think chronic inflammation, no doubt, is really important. We're still figuring out, it's sort of a catch-all term, and we're still figuring out the best way to assess chronic inflammation and specific types of conditions that develop from chronic inflammation. So that's sort of blood work. Maybe I've forgotten a couple of them, but I think that's kind of the blood work category. The other really low-hanging fruit is body composition. And so we do regular DEXA testing. Again, some people are worried a little bit about DEXA because of the radiation. In my view, fear of radiation in many ways is overblown in our society, and DEXA is really low radiation anyways. It's probably a flight from Seattle to California equivalent exposure. So why body composition? It's really important to know not only how much fat, adipose you have, how much lean mass, muscle in particular you've got, and bone density. Those are all really important. But it's important to know positional information. Where is the adipose located? And adipose is the most obvious of the three where positional information is important. It's particularly thought to be visceral adipose, the adipose around your organs in your abdomen, that drives a lot of the metabolic and inflammatory problems that people experience as they get older. The only way to really know, I mean, sure, you can sometimes kind of look at somebody and guesstimate. The only way to really know is to measure it. And so DEX is a super powerful imaging tool for understanding where is your fat located. Are you under muscled? Not too many people are going to be over muscled, but are you under muscled? And do you have any problems with bone density? Again, osteopenia, osteoporosis are huge problems in elderly people. And so the earlier you find out if you are on that path, the better off you're going to be, because you can take steps to avoid it. So I'd put that in the bucket that everybody should do. We also do, physiological assessments, VO2 max, grip strength, things like that. I think it's just important to know where you're at so that you can identify which people really need to focus in certain areas. We look a lot at, all of the 4 pillars, collect survey data from people. We do some cognitive testing to detect, cognitive dysfunction early on. Again, I think everybody should think about those things. Not everybody wants a super comprehensive health program, but I would put those in the areas everybody should be paying attention to. Your cognitive health is really important. And one of the things I'll say is there's a lot of fear, I think. People don't want to do these tests because they're afraid of the answer. I get it. Believe me, I get it. I've done a lot of testing. I've sort of gotten over that, but I understand that feeling, right? But information is power, and you can only take steps to address a problem if you know it's there. And the earlier you catch it, the better off you're going to be. So the only caveat to that is I do think there are a class of diagnostics that are susceptible to false positives, meaning it might give you a signal that there's a problem when there isn't. And the consequences of a false positive can be significant, either in the sense of significant anxiety or significant follow-up medical procedures. And so a good example of this, and I'm a proponent of whole body MRIs, but that's a good example of this, where for many people, a whole body MRI is going to detect an abnormality. It's going to be registered as potential cancer. And that's going to create anxiety in anybody, right? And sometimes will lead to additional imaging. That's fairly non-invasive. Sometimes will lead to a biopsy, which is more invasive. So tests like whole body MRI or multi-cancer detection, something like Gallery, the blood test, I put in that category. We do use those in our clinic, but we have conversations with people beforehand, make sure they understand this concept of false positives, what the likelihood's going to be, what it means if you get a false positive. It really, in most of these cases, means that you might have cancer, less than 50% chance still in most cases. So, and then we, through a conversational approach with the client, determine whether those tests are right for that person. No,
I mean, so I've had 6 whole body MRIs. I mean, I started doing this, like I mentioned earlier, years ago. And 2 out of those 6, I've had some sort of, one of them, ended up being nothing. I think there just wasn't enough data at that time, because now it's actually a known. They're like, oh, this is really normal.
Right.
And then another one that I had most recently, actually, it wasn't cancer or anything. So it can be a variety of different things.
Sure. Cancer is the one most people are going to think of, and that's probably the most common. But you're right, so it can be other stuff.
It wasn't even a signal of that, but it was showing something that, I'm in a very blessed position, right? So I got on the phone, called my doctor. I was in for another image, a repeat image, the same day.
Right.
And there was actually nothing there.
Right.
So that had just...
That was a true false positive, or it was just an imaging artifact.
Yeah. Literally. And so, and even, me, I have lots of resources in this industry, but of course I was concerned. I'm like, what is this? Like, what is that thing going on? But that is not the case for a majority of people.
That's right.
Being able to call, get in same day, have a quick analysis. Just so I could only imagine having weeks and weeks and weeks of going through these experiences, being concerned. So I think it's really important to go in with the right mindset, and it might not be for everybody.
Yeah. And that raises a good point, which is, again, something that I am a bit concerned about, which is the proliferation of companies that are making these diagnostics available, again, direct to consumer. There's value there. I'm not saying that shouldn't happen. I think there's value in making things available to people. And these are perfectly valid, really useful diagnostics. But the sort of middleman role of just facilitating people getting access, let's just say whole body MRI. There are people doing this with blood tests and with other things as well, without providing medical care, creates the situation that you just described, which is somebody goes and gets their MRI, they get an abnormal reading, and they then have to go to their primary care doctor, who then has to read that and refer them to a specialist. Yeah. And it can be, like you said, a 6-month process. Yeah. So I understand why companies do that, because it's actually really hard. It's really hard to run your own clinic and comply with all of the licensing and medical rules and laws. The healthcare industry is full of friction that needs to be fixed, but that's the world we live in. Yeah. And so they don't do that because it's much easier to just sell things and make money. But I worry that it's counterproductive and people are being harmed when you sell a medical diagnostic and you don't actually give people medical care, and you don't give them ways to get medical care.
Yeah. So because it's like a different business model, right? Like than what they're doing. It's like clinics are, as you mentioned, difficult, lots of regulations. So I get it, but no, I mean, it makes a lot of sense because I mean, health is the most important thing to the majority of people, right? Yeah. So if you find something.
It should be. I mean, I'm not sure it is. That's part of the educational thing, right? Okay. I mean, I think convincing, I agree with you completely, but I honestly, maybe among your viewers, that's by and large the way people think about it. But I think you get outside of the longevity bubble or whatever you want to call it. Typical person is not focused on their health. That's part of our mission, right? Is to change that, because you're right. Your health should be the thing that you prioritize the most.
But you're also right, because just look at the statistics, right? Like, Americans, 60% have at least one chronic disease, 40% two. Yeah. So you're right. I can definitely get a little bit of, and I'm sure you do too, right?
Yeah.
Because the majority of our friends are all like, doctors and they're physicians and longevity, connoisseurs or like advocates. So it can feel like everyone is on that same.
Yeah. It's really interesting you bring that up because you're right. I get that. I have to catch myself when I start thinking that way. And I'm constantly reminded, because I'm also in this really interesting place now where I interact with really accomplished, high-level people in different fields, very diverse fields. And I talk to them because they're interested in what I'm doing. I'm interested in what they're doing. And almost without exception, they have sort of heard about, longevity or MAHA or whatever. They're not paying any attention. They're not happy with their healthcare. They're still seeing a primary care doc. They're not doing anything proactive. Some of them are trying to eat a healthy diet, things like that. But the bubble is still pretty small. And so I get constantly reminded, and these are people, again, I'm not going to drop any names, but these are people who would be household names, big shots in politics or celebrities, right? Who haven't got the message yet. So again, that's all just to say, it's easy to point to the statistics for the typical American, but even people, high net worth people who should get the best healthcare in the world aren't. And so there's a lot of work to be done, which is daunting at times, but also means, that what we're doing is important.
Yeah. Yeah. I can think of an interesting example, but of course, like I don't really involve myself whatsoever in politics, but if, the past president immediately had this like massive cancer that has been so advanced, it's like, what's going on there?
Yeah. How can that possibly happen?
Yeah.
If it was you or me, we would have detected it years ago.
I know I'm doing like 500,000 biomarkers more than the president, apparently.
Yeah. That's a perfect example. Right.
Yeah. So, okay. So another test I want to ask you about is, I know that you're not a huge fan of supplements in general, in terms of their proven scientific widespread use.
I think that's fair. Yeah.
Okay. So what is your thoughts though? So a test that I do it's a micronutrient test. So what are your thoughts on that? Of course, it's an add-on, right? So it wouldn't be the first test that you would go to, but this is looking for, of course, I'm not running into deficiencies. I'm running some insufficiencies at times.
Right.
What are your thoughts on developing your supplement protocols based on a test like that?
I actually think conceptually, that's exactly what people should be doing, right? Okay. If it's something you can measure, and this isn't only for supplement deficiencies. This goes for the other biomarkers as well, right?
Yeah.
If it's something you can measure, and we know with some degree of confidence what the normal or even better optimal range is, then supplementing or taking medications to move the biomarkers into that range makes perfect sense. That's what we should be doing.
Okay.
The challenge is, I think, that it's a relatively small number of things that fall into that category. At least that's been my impression so far. Maybe you can educate me on this, but I feel like the number of nutrients, micronutrients that we can measure clinically, and that we have a high degree of confidence that there is this optimal range that's different from normal is relatively small.
Yeah.
So that approach is sound, and that's the approach I take. And that is a relatively small amount of the sort of overall supplement stack that a lot of people are using right now.
Yeah.
So it's when you get outside of that I start to be a bit more skeptical, a lot more skeptical. I would also say my problem isn't so much with supplements. Like, supplements, by and large, are bioactive molecules that can have both beneficial or negative effects, right? Mm-hmm. It's the industry that I have the problem with, and the bad behavior, at least in my opinion, of the industry writ large. Again, it's kind of like the biological age testing companies. There are good people with good intentions, and then in the supplement industry, it feels like there are a whole lot of people who just want to make money and they don't care if they're lying to people.
Yeah. Yeah. I mean, I agree with that. And I try to be really cautious on when people ask me what I'm taking. There are a few things like vitamin D, and look at your, omega index, and then omegas potentially, maybe some magnesium.
Right.
I put that in like this foundational bucket
I would
that
agree
probably
with that.
is not harmful, probably beneficial to most people.
Yep.
And then I always say and everything else that I'm taking, I also I'll talk about the longevity bucket in a second, but I then have my protocol supplements, which is all based on my own data. So I can't really give any advisement as to what anyone else should be taking, because they should get a test if they can, and then supplement what they need, because that's going to be the most efficacious way to do this. So
I agree. I guess a question I have, though, because you sort of separated vitamin D and omega-3 from the other ones, but it's kind of the same, right? You measure and you supplement to those levels.
I guess the difference in how I was thinking of it is the micronutrient test is going a little bit deeper versus just like a standard blood test. So totally, I mean, because you can take too much vitamin D, right?
Yeah,
like
absolutely.
if you are at 100 or 120, might be getting into concerning levels.
Yeah.
I like to keep mine somewhere between 40 to 60.
Yeah.
So, but I just try to make it clear for people, because people have questions, and I try to just, number one, always say that this is my data. This is what works for me. I also have been blessed enough to have, medical doctors, PAs, NPs on my team to look at what I'm doing, look at biomarkers, and then just share. And, too, when you're looking at thousands of biomarkers every quarter, there's always going to be something to work on. So I'm also not trying to, say that I'm like the healthiest person in the world. I mean, I'm in good health.
Well, apparently that title's already been claimed.
Yeah. Yeah. So, I mean, I do all the things, right? Like mastering the basics, but it's fun to just see once you've done that, like, because I'm ambitious. I love to like keep moving up. Like, what else can we do? So I think that test could be interesting. And then I do have like areas where I think it's interesting to explore, like potentially a precursor to NAD, like NMN or something. I also do testing. How much does that correlate to overall lifespan? We don't know.
Yeah. So let me ask you about the I agree with all of that. The NAD precursors, I've taken NR, NMN, NAD injection. So I've tried it all, right? Yeah. Again, mostly that was just to see if I felt anything, not because I really feel like they're likely to do a lot. But that's one where, you can measure NAD. Yeah. The challenge there is, I don't think anybody measures it well on the consumer market. So it's kind of like the biological age test. How do you know that it's accurate? And second of all, that's one where I don't think we know what the optimal range is, right? So it's a little bit of that's an interesting example. Let's just say you had a good test for measuring NAD. We don't really know what is optimal. And so you have to guess a little bit.
Yeah.
And let's be honest, you're guessing a little bit with vitamin D.
Yeah.
But we've got a lot more data there, and we've got large human epidemiological studies and a lot of decades of information. The NAD, we have almost no information to really tell us like what's optimal.
Yeah.
So there's also NAD could be its whole podcast. I don't want to spend a lot of time on it.
Yeah, and you've covered it a lot.
Yeah, I mean, there's all sorts of challenges there. But that's an interesting class of molecules. And conceptually, the goal there is to boost NAD. Yeah. The question is, what are you boosting it to? How do you know you're deficient? How do you know you're optimal? Does it make you feel better? Maybe that's the answer, right?
What about I mean, obviously, this is going to be a very tiny percentage of people that have access to this data. But if you have an NAD test from when you're 20 or 18, would you think there's any reason to try to maintain similar levels as you age?
I think that's not let me see how I'm gonna use maybe a double negative. That's not an unreasonable approach, right? Okay. So, and you can say that about a lot of things, right? If a biomarker changes with age, the intuitive approach would be to try to maintain it at the youthful level. So I think it's a reasonable thing to do. Is that a guaranteed recipe for success? No. Yeah. And there are situations where if you fix one thing, let's just say a hormone, it's working properly in the young state at that level, but the overall physiology changes as we get older. It can do different things in the aged state. So the concern people would have are things like, and again, I'm not saying this happens with growth hormone. That's a whole nother conversation, which is really interesting. But this is the concern people have with things like growth hormone or insulin-like growth factor 1, which is downstream of growth hormone. The youthful levels are great in the youthful physiology. In the aged physiology, particularly with IGF-1, there's concern that you might create cancer in that situation. Because, for example, your immune surveillance has declined in that case. And so the IGF-1 will seed cancers, your immune system will clear it, boom, all of a sudden you're in trouble. So it's not always going to be the case that supplementing to the youthful level is optimal. Yeah. It's just important to recognize that.
Yeah, that makes a lot of sense. Because even, I mean, theoretically, if you think about NAD, if it's improving the health of the cell, right, it potentially could increase the growth of.
I mean, people have said that about NAD. Again, I don't think that's a huge concern given the data I've seen so far, but it's certainly a hypothetical, and there is some reason to believe that could happen in some people. Again, NAD is really complicated. And I think, unfortunately, there's a lot of misinformation out there about NAD, even from well-intentioned people. Like there was just an episode with Eric Verdin and Peter Attia. Eric was on Peter's podcast. And I agreed with 95% of it. But they got into the NAD stuff, and some comments were made that as if it's 100% fact, like NAD declines precipitously with age. Like that's just not true. I shouldn't say it's not true. It's not established. Like that's a bit of a myth. There's some evidence that NAD levels decline with age. There's also evidence that it doesn't really happen, mice or people, at the population level. So it sort of gets presented by well-meaning individuals who are really smart and know their stuff as if it's a fact, and it's just not. Yeah. And so this idea that in everybody, or even across the population, NAD levels are declining in every tissue and organ precipitously with age just hasn't been shown. And so then the whole idea that we should raise NAD to fix that relies on something that might not be true. Yeah. So again, this gets to where if we had large-scale population data where we knew what the optimal level was, then we could supplement to that, and we had a good way to measure it. Yeah. And so this applies, I mean, NAD is an interesting case study of this larger idea that applies to all sorts of other molecules that we might want to take. Yeah. So it gets into a guessing game. And again, the way I think about supplements is the way I think about everything that I do for my health. It's a risk reward. And so how do you try to make a rational risk reward decision that the likelihood of doing this is going to be greater than any risk? And so you have to operate off of limited information. You kind of do the best you can. Yeah. I would encourage people to at least try to take that sort of a mental framework. Don't just take a supplement because your friend did it and said, they feel great. Don't just take a supplement because your favorite influencer said you should, right? Yeah. Try to do this real, risk reward calculation for yourself. I know it's hard. I know most people don't have the background in the space that we do, but I think it's a useful framework to think about as you're deciding what you're going to do, what you're not going to do, of all the possible things you could do or take. Yeah.
And I would just add, because I haven't talked about this much, but I think it's super important. And if you are going to take a supplement, I mean, please reach out, get their certificates of analysis and make sure that what is they're advertising the supplement is actually in the supplement.
Yeah, I mean, that's, again, yes, and that's easier said than done.
Yeah,
right. And you're trusting them. I mean, again, you're still trusting them to give you the real information. So this is where I think the industry problems are particularly profound, right? In that even if you have the best stack of supplements, right, you can be sidetracked because maybe the particular brand you bought doesn't have the level that it's supposed to have.
Which it seemingly
happens all the time. Yep.
Yeah.
Or there's contaminants in there that if you're taking 20, 30 supplements are going to build up in your body. Yeah. Exactly. So this is why I tend to take the approach. I want to be pretty sure that what I'm doing has a clear risk reward, favoring reward. It's likely to be beneficial. Optimally, I have a way to measure it, right? And I'm going to look at my sort of comprehensive panel of biomarkers longitudinally, maybe every 3 months, and monitor, right? And if something seems to be going the wrong direction, then I will evaluate what I've maybe changed in that time period so that I can take that out. And I also, I don't take very many supplements. Like, we talked about vitamin D, omega-3. I can measure both of those. I take that. Magnesium, even there, that's hard because you can't really measure like tissue levels of magnesium. It fluctuates, depending on fairly recent consumption. Yeah. Creatine, that to me is the no-brainer. Like, I think the data are clear enough now that for body composition, it's probably going to be beneficial, especially if you're doing resistance training, which you should be. Yeah. And now the data on cognitive function and, other potential benefits of creatine, that to me personally seems like a no-brainer.
5 grams, how much?
Yeah. So it's interesting. So I did 5 grams of creatine for a few years, and then I don't even remember why I stopped. I stopped for a while, and then I started again when the new data started coming out for higher doses at 10. And then I went and got just my sort of regular quarterly blood test, and my creatinine levels were through the roof. And so, it showed my kidney function, EGFR, as being like outside of the normal range, is low. Fortunately, I also get cystatin C measured, so I didn't get sidetracked by that. But when I went up to 10 grams of creatine, my creatinine levels in my blood showed an increase. And I hadn't seen that at 5 grams. So anyways, that's just maybe an aside for people. Yeah. If you're taking creatine and you get a blood test and your creatinine is high and your doc says you have early stage kidney disease, you might want to go get a different measurement of EGFR, and cystatin C is the way to do that. So 10 grams most of the time. Although, like, I've been traveling for the last almost 2 weeks, and I didn't bring creatine powder with me. So I'm less disciplined than most people who are really, really in this space, I would say.
Would you say, what does the research say for women? Is 10 grams also?
I mean, I don't know. I don't know that I can say with confidence that there is clearly a sweet spot with creatine right now. You probably would know more about the specific data for women. What I would say is, from what I've seen, 5 grams seems fine, right? And that at least for some of these other potential benefits, particularly in the realm of cognitive function, 10 grams seems to be where people are landing. Yeah. But that's an opinion that's very lightly held, and I don't know that we have enough data right now. I mean, what are your thoughts?
I mean, I've seen the new literature. I'm just curious, at what age would you want to start doing that for the cognitive benefits? Because now I'm doing still 5 grams, per day. Yeah.
I mean, I guess my view would be that going from 5 to 10, I think, is very unlikely to cause any problems other than maybe gastrointestinal. So, I mean, again, this goes back to individual. Like, if you start taking 10 grams of creatine and your stomach hurts, go back to 5 grams, right? Totally. I don't think there's a lot of reason to be concerned. On the other hand, there's not a lot of evidence that 10 is going to be a lot better than 5. So 5 is probably fine. Like, I don't think there's a wrong decision there. We just don't have enough information.
Yeah. Yeah, I haven't evaluated that in depth enough to make like a significant change. Yeah. And although, it's not a huge deal, you could just do 2 scoops, right? But still, it's going to change how I prep everything, et cetera. Right. So
the other one that might be worth commenting on, because it's gotten a lot of attention recently, and I'm really interested in it, is lithium, and particularly this lithium orotate formulation. And so, for a long time, lithium has been used clinically for psychiatric disorders at pretty high doses. And there's been this literature in the biology of aging that lithium, at least in invertebrate worms and flies, slows aging, increases lifespan. The mechanism's pretty well known there through GSK3 beta. And there's been this other body of literature in the dementia space in Alzheimer's disease mouse models with lithium showing benefits. And there was this new study out of Bruce Yankener's lab, just came out in Nature a few weeks ago, where I really thought it was pretty compelling story. So what they found was that in patients with Alzheimer's disease or mild cognitive impairment, there's localized depletion of lithium. They looked at a whole bunch of metals. Lithium was the only one that showed this. There's localized depletion in certain brain regions that are associated with Alzheimer's disease, where the plaques are most prevalent. And they were able to show that the amyloid beta plaques actually pull lithium out of tissue. So it binds lithium. So there's this localized depletion of lithium. And they were also able to look at human epidemiological data, and this has been known for a while, that lithium levels in the drinking water correlate inversely with risk of Alzheimer's disease and dementia. So there's human data correlative, and there's this interesting observation in human brains of lithium depletion. So then they went to mice and were able to rescue the mouse models of Alzheimer's disease, which again, some people would just completely ignore that. They're like, those models are terrible, don't pay any attention. They're imperfect for sure, but it's going the right direction at least. But the thing I thought was interesting was they found this specific salt of lithium orotate, which is what's called weakly dissociating. So you put it in water, it doesn't all become the two ions, the positive and negative, was resistant to being pulled out by the plaques. And so it's, again, far from certain, but suggestive that this may be a useful form of lithium to supplement with if you are at high risk for Alzheimer's disease, have mild cognitive impairment. The thing that makes me particularly intrigued is there's also this biology of aging connection. So it might actually be, I don't know if it's the same mechanism or a different mechanism. Presumably it's a little bit different mechanism, but there might actually be two things that supplementing with low dose lithium could be doing. So this is, I think, is an interesting example, because all of that I just said, you can probably tell I'm like kind of intrigued. I find it pretty exciting. I have not yet decided for myself if I'm going to start taking lithium orotate. I probably will, but I haven't decided for sure.
Yeah. It's interesting because that lithium orotate has actually been used in functional medicine for, I mean, I remember 6 years when I started my practice, they're like, oh, we can start, it was normal. So I'm excited to see that play out. So I've been on and off that for quite some time at a very low dose, obviously.
And part of the reason, it's not that I haven't added it to my stack because I'm concerned about it. Like, I think it's pretty safe, especially at, the low doses. I think it's at 5 mgs maybe. It's just that I have a pretty high bar before I make the jump. I'll probably try it and see. But the thing is, that's the kind of thing where it could certainly have a long-term benefit. You wouldn't really expect to feel anything.
Yeah.
So this approach that I'm guilty of taking as well, which is try something and see if I feel any difference, doesn't actually work for most things. For some things it does, but for most things it's not. You're not going to feel it right away.
Yeah. Yeah. I mean, same thing if you're looking at your labs and you're, suboptimal in nutrients or micronutrients, then you put those back, you're not going to really feel a big difference, right?
In most cases,
unless it was like significant.
Right.
But if they're just like, fudging with
but there at least you can measure it. So at least you get that dopamine hit from seeing the blood work come back, in the green range.
Yeah. Well, and I like have you done a micronutrient test with like
I haven't personally. I should. No, I should.
So it's kind of nice because it not only shows you, what you've recently been exposed to, but also a longer, kind of like an HbA1c. So, which is nice.
Right. And that I think is, this is sort of what I was alluding to, with magnesium versus vitamin D, right? So vitamin D, when you measure that, it's showing you sort of a level over the last weeks or months, right? And that's why if you're high and you stop supplementing, it's not going to go down right away. It's going to take a while. But other things that are water soluble, vitamin C is a good example. You can't really tell anything from that. That's telling you like your last 24 hours versus the last 3 months. And so you have to understand which metabolites fall in which category.
Yeah. I would love to go down the rabbit hole, but we won't today, of Alzheimer's and the research and what happened and where we're at now. But am I understanding it correctly that there was kind of like a big occurrence that may have tainted some of the Alzheimer's research?
Yeah. I mean, I think so there certainly have been allegations, and I think there's some truth to it, that there was some fabrication of some of the early data on the plaques and tangles really, driving the disease. I think, but this is a bigger, I think, challenge, right, with Alzheimer's disease research in general, or a criticism that gets leveled now, which is that the entire field for decades focused only on the plaques and tangles, and the idea that if we remove them, you're going to cure the disease. And now we know that's not true. That's sort of I'm not saying that, that's sort of the way it gets presented now. And so they were all bad because they all did this dumb thing and focused on the plaques and tangles. I mean, the reality is that was the logical hypothesis. That is a hallmark of the disease. And again, this is sort of the opposite of what we were talking about before. If you see something change with age going down, you supplement up. If you see something happening with the disease or with age, you pull it down, right? That's so it's logical to focus on that. Yeah. And I think it is the case that reducing plaque burden can have sort of modest, some might say incremental effects on disease symptomology. So my sort of take now is we have learned that the amyloid beta plaques and the tangles are a phenotype of the disease that are probably driving some of the symptoms that people experience, but they're probably at best secondary. They're not the primary cause of Alzheimer's disease. And some people would argue they're protective, that what these plaques are doing is actually pulling the toxic components out of solution. I actually think Alzheimer's disease and dementia in general is probably much more complicated even than that, in the sense that there's this underlying inflammatory component, neuroinflammation in particular, that is what's in most cases leading to the neuronal dysfunction and death. Yeah. And that inflammation can be caused by lots of different, maybe not lots, by multiple different types of signals, right? Yeah. And so I think the soluble oligomers of A beta might be one, the plaques themselves might be another, bacterial antigens that get to the brain might be another, mitochondrial dysfunction might be another, heavy all metals, of those things. Maybe. Yeah, metals in some cases. Yeah. Those all can drive, metals could probably have toxicity for a few different reasons, but they all can contribute to inflammatory burden and or to neurodegeneration. And then what you see, depending on where that's happening in the brain, you get these different classes of dementia, right? Yeah. That's what drives the way the disease presents. Yeah. And so one piece of good news there is, if there really is this core commonality at some level that's driving multiple types of dementia, we have the opportunity to target that directly, as opposed to targeting only Alzheimer's disease or only, frontotemporal dementia or other types of dementia. So that's still a hypothesis or an intuition maybe, but I think there's pretty good reason to believe that there's some grain of truth in there. And, if you wanted to take the counter position to the lithium case that I just presented, based on the new paper, you would expect lithium to only be effective in conditions where amyloid plaques are a big component of the disease. That model is that it's those plaques that are pulling lithium out of tissue, right? Out of specific regions of the brain. Time will tell. The thing that gives me a little more optimism is that lithium also seems to affect the biology of aging and has effects on brain chemistry in people who don't have Alzheimer's disease, right? So there is the potential it could be more broad if it has protective effects against dementia.
Yeah, I love that. Obviously, we are both fans of mastering the basics of health optimization, sleep, exercise, everything that you mentioned in the beginning of the podcast, social connection. What would you say, if someone was just starting out, would be the biggest lever to pull first?
It's going to be individual. So I think this is partly one of the exercises that we go through anytime we have a new person come into one of our programs. We look at the 4 pillars: eat, move, sleep, connect, right? And try to figure out for each person where they at, right? So for some people, cleaning up their diet is going to be the biggest lever they can pull. For some people, starting to exercise regularly or getting on an exercise regimen that's appropriate for them. For some people, it's going to be sleep. And for some people, it's going to be their relationships. And I wouldn't say you have to only fix one, right? Like, I think that's one strategy. Identify where you're weakest and really lean into that. But I think it's going to be different. So I guess if I was to look at the average person, well, I was going to say I can look at the average person that we see, but I don't think our clientele are typical, right? Of typical cross
They're
section.
already doing a lot.
I guess what I would say is, so we actually have a program that is geared towards employees, right? So employees of a large company. It's a pretty good cross section of at least, Pacific Northwest, middle class America, Pacific Northwest.
Yeah.
So there, I can say with confidence, we see a lot of early stage metabolic disease, where certainly, sleep can be a component, but in that case, nutrition and exercise are really the levers you're gonna get the biggest effects from right away. So the five, so I talk about the four, right? Right, nutrition, exercise, sleep quality, and connection. Connection, I think it's worth saying, we encompass both sort of like your own personal connection, mindfulness, some people would call it, and human relationships in that bucket, even though they're not identical. So those are the four. The other one, I think, that falls out of the pillars of health span, but where a lot of people in our clientele, and so we're talking, 40s, 50s, sometimes 60s, can get the other big lever is hormones.
Yeah.
That's where if there is dysregulation and you fix it, you have a huge impact.
Yeah.
So I would say those are the five really that can have the largest impact for most people.
Yeah. Yeah, I totally agree. Something that I've really seen as a woman is like stress. I mean, stress for everyone.
That cuts across all of them. Yeah.
For everyone. But, it's interesting because I can look at my data and it's just me and my husband, but also there is, a sentiment out there that women report significantly higher levels of stress. Not saying that we're more stressed than male counterparts, but just how we perceive it. And, before we started the podcast, we were talking about how women's brains work. That could be definitely a part of it. But I've just rolled out this protocol. So I'm trying to reduce my stress. I'm measuring in an Oura ring. But I don't think I've ever seen anyone actually put out a protocol for that and share the data. But it's been really fun implementing some interventions because stress is, I think, one of the hardest pieces because it's not just like a checkbox. It's like you did your workout, you ate your food, and you spend time with friends. It's like something is going on. So I'm excited to see how much progress I can make on that.
Yeah, I mean, and I think I get this question a lot. Shouldn't stress be one of the pillars? And I think that's a reasonable way to sort of formulate things. The reason why we chose not to do that is because we really feel like stress cuts across all of the pillars of health span, both ways. Meaning that if you have a high level of stress, that can impact your eating habits, that can impact your willingness, ability to exercise, certainly impact your sleep quality,
of course,
right? Yeah. And it can impact your relationships or your ability to, be at peace, have a sense of purpose, your mindfulness. And improving any of those things can have a positive impact on the way that you perceive stress. So, and stress is sort of another catch-all term. It's kind of like inflammation, right? Yeah. There's a lot that goes into that. And some stress can be good, just like some inflammation can be good. Yeah, so totally. I think that's one of the challenges why you haven't really seen any, at least really science-based approaches to protocols around this or measurements around this is because stress is a bit murky what that actually means. And so we can measure some of the consequences of stress. Yeah. We can measure some of the hormones associated with stress, but we can't actually measure stress per se.
Yeah. I mean, you could get a snapshot of cortisol, right? But I mean, we also know that you can be physiologically feeling stressed and your cortisol levels can remain the same. So it's very difficult, but I think, I just completely agree and love the idea of everyone repositioning because everybody just wants what to do, right? But it's like you have to really reflect internally and look at your labs and see where do you need to start. And then that's going to be the biggest lever for you.
Yeah. And I would also say, I mean, I think a lot of this is very personal because for me, one of the things that I find is most effective at alleviating stress is getting out in nature and doing fun stuff with other people, right?
Totally.
So I'm very much one of these people who I feel like, and again, I'm not particularly rigid in my approach to health, right? Yeah. That if I was to go down the path of really trying to map everything out and do everything perfectly, that would create stress for me.
Yeah.
Which in the long run would be detrimental to my own health. Right. Some people that works really well for. So I think you have to understand your own approach to life and what brings you joy. And because that's going to feed into the way that you manage and respond to stress. Yeah.
Yeah, absolutely. I agree with that. What is what is known to you between the differences between male and female aging?
Yeah, well, I mean, I think there's some obvious things. Women in general live longer than men, pretty much across the board in developed nations. We don't really understand the biology there. There have been some hypotheses, but there is it's talked about that women have shorter health span than men. That's a little bit harder to actually quantify. This is maybe worth saying out loud. We have no way to measure health span. There is no consensus definition of health span. I think we all kind of get it intuitively what we mean, but we don't have any way to measure it. And so when you hear people say women have shorter health span than men, but they live longer, that you can't actually measure that. I think you can say women live longer with some chronic diseases than men do. And so that's one way to estimate health. But I think this question of whether women are healthier than men in their 60s and 70s and 80s is harder to answer. The biggest obvious difference is women experience menopause. So both men and women have hormonal changes as we age. But in women, first of all, it's 100% penetrant. As far as we know, every woman that's ever existed and lived old enough has experienced menopause. It's 100% penetrant, and it happens in a shorter window of life than the hormonal changes that men see as we get older. There are significant consequences to those hormonal changes that go along with menopause. And I think that it's become clear to me, and I think most people in the space would agree that those hormonal changes that happen during menopause are what drive some of the increased risk for chronic diseases that women experience compared to men as they get older.
Yeah.
And so that menopause process, which is caused by ovarian senescence and ovarian aging, is, I think, a really interesting space for biological research into what the processes are that lead to ovarian senescence and how closely are those connected to the underlying biology of aging. I think right now it looks like they're pretty intimately connected. That would certainly be my hypothesis. And so that means that by understanding ovarian aging, we're going to learn something about the biology of aging. And the flip side is some of these interventions that modulate the biology of aging might have a positive impact on ovarian aging and delay menopause. So there are a couple of directions we could take this. We could either talk about that biology or we could talk about hormones. They're both super interesting, super important. I'll leave it to you to see which way you want to go.
Yeah. I mean, I'm a huge fan of bioidentical hormone replacement.
Yeah.
So when I get to that place in my life, I will totally be all in on that.
Yeah. That might just be worth double clicking on. And I know, again, probably most of your listeners will already have heard this, but a lot of people still haven't. That, the idea of hormone replacement therapy, bioidentical hormone replacement therapy, or hormone therapy, was sort of derailed for a whole generation by pretty much a single study. It wasn't a bad study. It was misinterpreted. A lot of unfortunate hype around the interpretation that hormone therapy causes breast cancer. That now seems to be completely wrong, at least when done up to current standards. Yeah. And there are huge benefits for many women that go along with hormone therapy. And so I've heard Jennifer Garrison, who I know you know, say this, and I completely agree. The question, in my view, for a woman in menopause when she's talking to her doctor shouldn't be, is hormone therapy right for me? It should be, is there any reason why I shouldn't do hormone therapy? Like, why would this be wrong for me? I think that needs to be reframed. And I still hear people with large followings refer to the Women's Health Initiative study and say hormone therapy causes cancer. Like, it's really unfortunate.
Can you DM me those influencers? I'll send them, because that's just incredibly harmful.
I agree. I agree completely.
And then that's so sad. They're already, I mean, I think the stats are abysmal in terms of how many women are actually even on hormone replacement therapy.
It's like 5%.
Something about 5%. Exactly. So that is concerning, when we know that there can be this huge upside. There's some good rumblings going on. So a lot of female docs recently met with the FDA because there's this black box warning that is, unduly put there, which is obviously concerning for the doctors prescribing it, nor are they even necessarily trained on how to prescribe this because of this permeation of misinformation,
Yeah, I think
20
what you
years
just
ago.
said is really important. Because of the way the Women's Health Initiative was interpreted, a bunch of women who were on hormone therapy were
Yeah.
stopped.
Yeah.
And a whole generation of doctors they didn't get
Yeah.
this biology in medical school.
Yeah.
Right? They don't know anything about menopause. Well, I shouldn't say that, but at least the approaches for hormone replacement, right?
Yeah.
And so it's frustrating to me when you still see primary care doctors who refer back to the Women's Health Initiative study as their source of information on this topic. And you understand that they never received any training, so they feel uncomfortable with the topic. That's not a valid excuse not to get up to speed, but that's I think what leads into this a little bit. So there's work to be done, but I agree with you. The momentum is shifting, and it's still very frustrating how much lack of understanding, misinformation there is out there, and that the numbers still are as low as they are. Yeah. So hopefully that will change. One hormone in there, though, you mentioned the FDA warning. It seems absurd to me still that there is no FDA product for testosterone for women.
Yeah.
So that's another place where, I think a lot of times, even people who sort of know about hormone therapy for women, they think about estrogen and progesterone, but they don't think about testosterone, which is super important in this space as well.
Yeah.
And again, I'm a man. Maybe I shouldn't even be talking about this, but I've experienced this, in my own life, how much benefit can come from for women who are experiencing symptoms of menopause. And hopefully the news will get out there that it's time to get on board with the science.
Yeah, no, I mean, I agree. It's also a bit odd to determine that you should in fact supplement 2 of the hormones, but then you leave out, another primary hormone. So even when I was running my clinic, I mean, all the time women would come in and be like, "Oh my gosh, you guys prescribe testosterone here? This is so hard to get."
Yeah.
And that's so sad.
Yeah. And it's interesting to me. I mean, I think this is a kind of case example of a phenotype that I've seen more than once, where you have women who are going through menopause. They get the estrogen and progesterone supplementation, and they have a certain phenotype. I'm just going to say it that way, where they are very lean. Mm-hmm. You would look at them and be like, this person's in perfect health, right? They're very lean, usually active, and have metabolic dysfunction. Mm. And if you do a DEXA, you see they're under-muscled. So they're lean, but they're under-muscled. There are well-established metrics if you measure by DEXA, which is what I think most people should be doing, for what the healthy ranges are for lean mass, bone density, and adipose. Now, the adipose, as we've already alluded to, you want to look at visceral adipose and whole body adipose, right? So those are the metrics that I would use in general for assessing whether somebody is an optimal, normal or suboptimal. I think you could have a difference of opinion where you want to target people, because those are population averages. I would say for most women who really want to be as healthy as they can, and this is probably true for men as well, being somewhere in the top quartile, highest 25%. I don't think with lean mass, again, I don't think you're going to become over muscled. And this is a myth. I think that unfortunately some women are hesitant to take testosterone because they're worried that they're going to, blow up into a fitness model or a bodybuilder. That doesn't happen. Yeah. It doesn't happen. You really have to work hard for that to happen. So it's not going to happen accidentally. Yeah. Totally. But going back to this case example, the metabolic dysfunction is being driven by being under-muscled. Mm-hmm. And being under-muscled is being at least partly driven by testosterone deficiency. Yeah. And so if you fix the testosterone deficiency and the woman does resistance training, you can then increase lean mass. That'll become a sink for the glucose, and that'll at least improve the metabolic dysfunction. I
love the term that muscles like a metabolic Spanx. So Spanx is something that women wear. Do you know this? But it's I love the like visual in your brain, right?
It's so important. So I think, it's hard to imagine having too much muscle. Certainly it can happen, but you'd really have to go overboard to achieve that. So that should be a place where everybody, men and women, should be prioritizing.
And having, I usually keep my body comp around, I think I'm about 87% muscle. So it's high, right? But for me, I mean, but I don't look, I
Well,
don't think I
let's
look
clarify
like
that. You're 87% lean mass.
lean mass.
Yeah.
Yes.
So
yes,
lean mass
thank
is
you.
going to be made up of water and
Yeah.
other stuff as well. But
Yeah.
but I get the point.
So curious of your thoughts on this too. So I'll do DEXA like once a year, but then at home, I just do a bioimpedance scale,
Yeah.
like, maybe once or twice a week,
Yeah.
because I feel like, the numbers aren't extraordinarily off, but
Yeah.
it also, I think, is energizing, right? When you see changes in fat mass and muscle mass. So
Right.
I like doing that. Do you
Right.
have any issues with those types of scales?
I don't have any issues other than recognizing that they're going to be less precise
Yeah.
than a DEXA, right? But I think my experience has been, in general, the bioimpedance reports body fat lower than DEXA.
Mhm.
But I think if you do both, and that may not be true across the board, this has just been my personal experience
Yeah.
and what I've seen with other people, may depend on your body composition and your device. But I think if you normalize the whatever bioimpedance device you're using to your DEXA, and then
Yeah.
looking at changes over time, that's a perfectly reasonable approach.
Yeah. Yeah. Well, great. So one other question as it relates to, we were talking about female longevity. So I've dabbled in a little bit of rapamycin for that specific reason, but I know that you have, done a ton of work on rapamycin. Can we just touch on like longevity drugs, such as rapamycin and metformin, what you are excited about with them?
Sure.
And then maybe touch on to this Vibrant study that is not yet peer reviewed, not yet finished, but has some exciting, exploratory evidence so far.
Right. So I won't get too far in the weeds on rapamycin unless you want me to, but I think in general, the concept here is going back to the biology of aging. There's a network that underlies the biology of aging, the hallmarks of aging, and there are ways that we can tweak that network that seems to slow aging in laboratory animals.
Mm-hmm.
And rapamycin is a drug that does that. Among drugs, it is the one where we have the best evidence in laboratory animals that it consistently, reproducibly, and robustly slows biological aging. And the way we measure that is by effects on population lifespan and then multiple health span metrics. And rapamycin across the board increases lifespan in mice 15 to 25% on average, and seems to delay pretty much every age-related functional decline in disease, as you would expect if you slowed aging. That is kind of the gold standard in the sense that it has been shown the most times and the most comprehensively, both across different strains of mice and different health span metrics. Then there are a set of drugs that have been tested by something called the interventions testing program that people would point to. And I'm starting with the laboratory animal stuff because we have to be honest, we don't have anything that has been proven to slow aging in humans. It's just a fact. Other than, I would argue, you could make a case for these lifestyle factors. Mm-hmm. So the other drugs that people will point to are tested by the interventions testing program, which has also tested rapamycin in mice. This is an independent NIH-funded program that tests drugs at 3 different sites in parallel. So it's triplicate replicate. So there are, depending on your criteria, between like 9 and 12 things that have come out as extending lifespan there. The ones that most people are excited about, talking about hormones, 17-alpha estradiol and estriol, so both estrogens, have been shown to increase lifespan, predominantly larger effects in male than female mice, which is interesting. Yeah, we don't understand it, but I'll get to the human data in a minute, which is also interesting there. Acarbose, which is a molecule that acts in the gut to impair breakdown of complex carbohydrates into simple sugars, so it probably reduces glucose uptake. A class of drugs called SGLT2 inhibitors, canagliflozin is the one that came out of the ITP. These are really interesting drugs. What they do is they inhibit a protein called SGLT2 in the kidney and cause you to pee out more glucose. So they're approved anti-diabetes drugs.
What if you just keep your glucose stable all the time, though?
Right. So this is a different question you're asking, which is these drugs, like that's a good case where we know the mechanism of action. If you've already optimized all the other stuff, will these drugs have any benefit? That's a different question. We don't know the answer. It's a good question. My intuition is it depends on the drug and the person. But you're right. An SGLT2 inhibitor for somebody who's got their glucose dialed in between 80 and 100 all day, probably not going to do much more benefit there. Might even do harm because you bring your glucose too low. Nonetheless, that has been shown to increase lifespan in mice. And there are another handful that are potentially interesting. You asked about metformin. Metformin's an interesting case where it was originally reported to increase lifespan in mice, but that was in a specific strain background that got a lot of cancers, was very short-lived. The ITP was not able to show lifespan extension, and the effects that was shown in one other study were tiny, like 3%. Mm-hmm. So metformin is not, from a biology of aging perspective, not really near the top of the list of drugs that seem to have an impact on the biology of aging in mice. Okay. So then we can look outside of mice, and that's where metformin gets a lot more interesting. Metformin, very well-known, very effective diabetes drug in people. If you look at diabetics taking metformin and you compare them to diabetics not taking metformin, the people taking metformin live a lot longer, even diabetics who are taking other drugs. Mm-hmm. It was originally reported that diabetics taking metformin had lower mortality, which you would expect to equate to living longer than non-diabetics. That has since been debunked, or at least it hasn't been reproducible in a couple of studies. So I think that was just wrong. Mm-hmm. So where we're left with metformin is it's a really good diabetes drug. We don't really understand its mechanism of action. There are hints that metformin may reduce the risk of other age-related diseases, dementia, cardiovascular disease, kidney disease, which is what you would expect if it was slowing aging. But that's all in diabetics. Mm. So I am of the opinion right now that metformin for non-diabetics is not a great bet. It's pretty safe, but there are some side effects that are becoming more understood. The two that I personally, that keep me from taking metformin, are lowering testosterone in men, some men, and evidence that metformin may blunt the benefits of exercise. So again, that hasn't really been proven, but there's lines of evidence supporting that. So to me, the risk reward for metformin, if you're not diabetic, probably doesn't favor reward. The interesting human data that intrigues me outside of the rapamycin off-label use, it comes from a study where they looked at the UK Biobank data, where they were able to ask the question, if you look at all the prescriptions in the UK over some time period, and you compare people taking a given drug to people with the same condition or the same health status not taking that drug, what are the drugs where there are differences in mortality and risk of death? And 7 or 8 popped to the top of that study. Some of them overlapped with the interventions testing program. The most interesting to me out of that group are the estrogens and the SGLT2 inhibitors. So this is a case where you've got admittedly correlative, imperfect human data and really, really solid mouse data, but it's in mice, but they're pointing the same direction. I find that interesting. You would not expect that to happen if there wasn't some underlying biological mechanism there. So those are classes that I look at, and I feel like there's pretty good evidence that these may impact the biology of aging in people and have broader health benefits.
Yeah. Any thoughts or comments on this potential slowing of ovarian aging with rapamycin?
Yeah, I'll talk about that in a minute. Let me say the other class of drugs that I am also intrigued by in this space are the GLP-1 agonists, right? So, and there are 2 ways that a GLP-1 agonist could slow aging. One is from a simple caloric restriction mechanism. And I think we can make a pretty compelling argument, again, not rock solid, but pretty compelling argument that food consumption across some range is correlated with faster biological aging, right? People who are obese, particularly if they have metabolic disease, in many ways appear to be aging biologically faster. And people who are very metabolically healthy and thin seem to be aging slower. And we can point to the mouse data where caloric restriction is the gold standard intervention for slowing aging in mice. So the GLP-1 agonist could be slowing aging through caloric restriction. That's kind of okay. I mean, it's cool in the sense that, it's a useful medication for people who need to get to the optimal body weight. But I think there's also reason to think that the GLP-1 agonists are having effects independent of appetite suppression that could be modulating the biology of aging. This is very speculative. There's no real rock solid data yet, but there are intriguing sort of hypothetical ways this could be working through inflammatory mechanisms that GLP-1 agonists might actually slow the biology of aging outside of the appetite suppressing effects. So I'm intrigued by these drugs both for both reasons. And I think we'll obviously, as a society, continue to pay attention to them, but it'd be interesting to learn if they can have broader effects on health. This may also, sorry, I'm derailing us from the ovarian conversation, but this may also be a good time to talk about where even if an intervention slows biological aging in people, it might not be a win in terms of something you want to apply in your own life. I think caloric restriction and maybe the GLP-1 agonists, although there are things we can do there to mitigate this. Caloric restriction may be an example where the amount of caloric restriction that optimally slows biological aging is so severe that it is going to have detrimental effects that offset that benefit of slower biological aging. Again, I can't prove caloric restriction slows biological aging in people, but there's lots of data supporting that. It seems like a pretty good bet.
Yeah. And
we know that severe caloric restriction has psychological consequences, social consequences, and leads to frailty. And so it is my speculation that the optimal level of caloric restriction, if you're purely looking at rate of biological aging, is actually going to lead people to die earlier because of these other consequences. So it's just a useful thing for people to appreciate. Rate of biological aging is not the be-all, end-all of health span or lifespan.
Yeah. Well, it's a great point because, when I first started doing all of this, I was implementing these very science-backed practices, right? I'm a woman. I was in my late 20s at that point. Now I'm in, I'm 34. So what I found is that my thyroid health started to decline. My hormones became dysregulated. And at the stage of where I am in life, I don't want my hormones being all over the place. So, that was one of the big things that really prompted me to dive deep into what works for me, not only as an individual, but also as a woman, because these things might differ.
Yeah.
Whereas some individuals in the space that are men might get away with doing that, but, might not be applicable. We know nothing is applicable to everyone in every situation, but that was a big, aha moment.
Yeah. And I mean, that's a case where, going back to what I mentioned about the estradiol and estriol in mice, at least in mice, there is this clear situation where some drugs extend lifespan robustly in one sex and not in the other sex. And so it is very likely to be the case that there will be interventions in people that have that similar sort of, sexual dimorphism. And we don't understand that biology right now.
Yeah.
So worth just being aware of.
Absolutely. Yeah. I tried a GLP-1 for a very short, I was doing 5 units of tirzepatide, but it suppressed my appetite so much that, to be honest, I didn't really need to lose, I don't want to lose weight anyway. So, and then resting heart rate was up a bit. So yeah, I was excited about it, figured I would try it. I only ended up staying on it for 2 weeks. But this is kind of funny. And I don't think I've ever said this before, but I've actually gifted a few vials of tirzepatide to individuals, or different GLP-1s. But I think if you need to lose weight, man, I mean, I would be all over that. I'm excited to see what happens with the data and any potential side effects, but I think it seems pretty exciting.
Yeah, I would agree. And again, the drugs are extremely powerful, have a lot of upsides. There are some concerns appropriately. Now people are starting to recognize the risks around loss of lean mass when you lose a lot of weight rapidly.
I mean, for sure, if you're on those, you should be strength training.
I agree completely. Unfortunately, when they were first, sort of rolled out, the physicians weren't recommending that. And so the early studies showed significant lean mass loss. And so they have some downsides that can be mitigated when they are used appropriately in the context of overall lifestyle and potentially other medications. But I agree, the benefits for lots of people are huge. And, it's kind of frustrating to hear a little bit of demonization of these drugs. It's no drug is perfect, but these drugs do a lot of good for a lot of people. And I think, we're going to learn more about the long-term benefits and potential consequences because they haven't been around that long in use to this extent. But I think clearly for a lot of people, the upside is significant.
Yeah.
Okay. So ovarian aging. Yeah. So again, this goes back to this idea that, ovarian senescence in women is, one of the first major hallmarks of aging that women experience from a functional perspective. And the hypothesis, which I think is pretty clear now, is that ovarian senescence is influenced by, when that happens, is influenced by the rate of biological aging of the woman, right, prior to that. And so the underlying biology of aging, much like it contributes as a risk factor to dementia and different types of cancer and heart disease, is driving at least the early processes of ovarian senescence. Now, this might be getting a little wonky on the science side. The one place where ovarian senescence is a little bit different from these other age-related conditions is that this has been something that evolution has selected for. And I don't think we really completely understand why evolution has selected for ovarian senescence and menopause in humans. There are lots of hypotheses out there. And the reason why that's important is that the other age-related diseases and their association to biological aging could be somewhat different than ovarian senescence because there's been this evolutionary pressure for this to happen in this compressed window of life. Nonetheless, what we are starting to see is that interventions that seem to slow biological aging or modulate biological aging can change when ovarian senescence happens, at least in animal models. And in some cases, depending on how far into the ovarian senescence process, actually reverse it. And so that's where rapamycin, I think, is really intriguing, is there are multiple labs now that have shown that you can take mice where their ovaries have become dysfunctional. They're no longer fertile. You can treat them with rapamycin. You can restore ovarian function and they can have pups. And this is a normal age-related process in mice, much like it is in humans. So there is now obviously a lot of interest in the possibility that rapamycin, or potentially in the future other geroprotective compounds, could be used to delay or reverse ovarian senescence, menopause in women. There's a clinical trial being done out of Columbia. This is Zev Williams and Yushin Su, looking at whether rapamycin can be effective in women who have premature ovarian failure. The clinical trial is still going. I don't believe any results have been released, so I can't comment on the outcome of the trial, other than to say I think it's pretty clear that within that trial, and we have lots of other data now from both other clinical trials and off-label use, that rapamycin can be used safely in otherwise healthy people of normal health status, without a lot of the side effects that people were concerned about based on rapamycin's approval and use as an organ transplant drug.
Yeah. Well, will you keep me up to date if you see any more
I sure
female
will.
longevity?
Yeah. Yushin's
info?
a good friend of mine, and so I try to press her periodically to give me some of the data. But if the study's still blinded, she can't really do that.
Yeah. Well, we're all excited to see what does come of that, because that would be amazing. Well, I mean, we could have a podcast for another 5 hours because we've really just started to scratch the surface, but this has been such a pleasure. Where can people find you? Yeah.
So I am on, what am I on? I'm on X and Instagram at Kaeberlein. And we have our own podcast called the Optispan podcast. So you can definitely see all you want of me there.
Love that. Well, we'll make sure to include everything in the show notes. And thank you again for coming on.
Thank you.